Welcome to the Pod of Inquiry, pushing the envelope for human understanding and optimization, the podcast for podiatrists. The Pod of Inquiry is designed to empower you with knowledge. What happens from there is up to you. Your host, Dr. Stephen Barrett, has designed this show to take you down some very deep rabbit holes, hopefully bringing you back out again, relatively unscathed, but cerebrally whipped, enabling a better understanding of all things worthy of inquiry. If you have more questions after the show, then that is good. The new discovery today many times was the new discovery 50 years ago, only to be suppressed or plainly ignored. Medicine and surgery can sometimes take a long while to get their paradigm shifted. We hope to have a lot of fun on this show and maybe destroy some ridiculous dogma along the journey. Thanks for joining the show today. Let's start spelunking.
In this second episode of the Spelunker Summer Break series, we're delving into the fascinating world of stem cell science again and its potential to revolutionize regenerative medicine. Our guest is Christian Drapeau, a renowned stem cell researcher and the pioneer behind Stem Regen, a natural compound that's been making waves in the field of stem cell nutrition. Christian's work focuses on harnessing the body's own regenerative capabilities to promote health and combat aging. Stem Regen is a unique blend of natural ingredients designed to support the release and circulation of adult stem cells from bone marrow. This approach represents a paradigm shift in how we think about supporting the body's natural healing processes. In today's episode, we'll explore specifically the science behind Stem Regen and how it works to mobilize your own stem cells, the potential applications of this technology in various health conditions, the broader implications of regenerative medicine and anti-aging research, and the journey he faced from concept to product. We'll also discuss the latest research in this field and what it could mean for the future of healthcare. I can attest that I have personally taken this supplement now for more than 1 year, as well as recommending it for all of my surgical patients to help them heal faster. And it is shocking to see how impactful this agent is for regeneration. Please enjoy this conversation I have with Christian Drapeau. Well, I'm here with Christian Drapeau, and Christian, thank you so much for taking the time to join the show today and share your expertise on what I think is an absolutely fascinating subject. We're going to talk about the mobilization of endogenous adult stem cells, and I've got your book right here, which I was telling you earlier before we jumped on the recording of how much I liked that book because it was just a great treatise on really pretty much all things stem cells. So thank you for taking the time to write it, and thank you for your expertise in writing it in such a great readable format. So let's talk about
Thank you so much.
You're welcome. And so let's talk about how you got to the stem cell arena. I know you started out as a neurophysiologist up at McGill, right?
Correct.
Okay.
So I was doing research. Yeah, go ahead.
Well, I was going to say, so you obviously there had to be some event that took you from the neurophysiology world into the adult stem cell world.
Well, I was in that field, and there's a product that was on the marketplace, blue-green algae from Klamath Lake, also known as AFA. Its scientific name is Aphanisomnon flus aqua, abbreviated AFA. And and one of the benefits of AFA was its effect on the mind. People were reporting that it was increasing mental clarity, mental energy, to a point of being antidepressant. So partly for that reason, partly also the company needed somebody to lecture for their product. And in my prior years, I had done a lot of work on accelerated learning, how to learn fast, how to teach so people can learn fast. And you combine these two, and I guess I was a good candidate for that company to head the research on that product. So I was hired by this company in 1995, and my role was really to document the mechanism of action behind the health properties of that product. So the main benefits were an effect on the immune system, on inflammation, and on the brain. So very quickly, we identified that the product was contained phycocyanin, which is a COX-2 inhibitor. It's present in all blue-green algae. It has a polysaccharide that supports the immune function, specific aspects of the immune system. And it contained phenylethylamine, PEA, known in chemistry as the molecule of love, the molecule of joy. Whenever you feel content, your brain is making PEA. It potentiates noradrenaline and dopamine transmission in the brain. So as we do all this work, we really explained or documented the mechanism of action behind the benefits of AFA. But as I'm doing this, I came across cases of people who reversed multiple sclerosis, severe heart disease, Parkinson's, severe emphysema, Alzheimer's, liver failure. So you're looking at all the spread of these conditions, and it was for me like a mystery. Like, what is this product doing, leading to benefits in some people to the heart, other people the liver, the pancreas, the lung, the skin, the brain? From a scientific standpoint, products do one thing, maybe two, but they don't touch all aspects of human health. So we did not really have an explanation until early 2000, actually early 2001. I came on an article that was describing for the first time, to my knowledge, and I think in the scientific literature, how stem cells from the bone marrow went to the brain and became a neuron. We need to understand two things. In those days, stem cells are only known to be precursors to blood cells, and of all tissue, the brain does not regenerate. So to me, it was like a really amazing report. So I went to the local library, started to search for other articles like this one in the scientific literature, and I found a study documenting how stem cells could become heart cells and liver cells. So now you put yourself in my shoes, early 2001, at a time when stem cells are only known to be precursors to blood cells, and you come across 3 studies showing that they can become heart, liver, and brain. So the question was, what could be the mechanism of action for them to become heart, liver, and brain, but not pancreas, lung, skin? They have to be able to become the rest as well. And if they do, then they have to be the repair system. How can you see a stem cells able to become all kinds of cells in the body, but it's not its function? It has to be its function. So I published an article in the journal Medical Hypotheses, suggesting that stem cells were the repair system of the body. And in the back of my mind, the thought or the hypothesis was, what if this plant, AFA, its main mechanism of action was to trigger the release of stem cells from the bone marrow? It was not a thing at the time, so nobody had ever looked at this. So we acquired a flow cytometer, we started to count stem cells, and that's what we discovered. And the idea to me was honestly, when I had this product releasing stem cells and everything that was at the time available but not yet fully documented, I looked at this and I thought, we've just discovered a way to put an end to disease. In my mind, that's what it was in 2001. And of course, we're 20 years later right now, there's tons of science. It takes a lot of time for that concept to trickle into mainstream medical knowledge, if you want. But that's how I started. The moment that I discovered this, then I dropped everything else and I went into stem cell research.
So you made a nice point at the beginning of your book where you talk about wellness is the ability to regenerate more than you're degenerating. Correct. And that chronic illness or sickness is basically your inability to regenerate more than you're degenerating. And so, I think one of the things that you make such a great point of is that we have these adult stem cells that are resident in our red marrow. And I guess we should define adult stem cells because they're adult stem cells at about 4 or 5 days after birth.
They're into your body. Yeah, before that, they are embryonic, then fetal, and then so-called umbilical. So they're there in your bone marrow.
So so you have these, and just from a clarification, I think a lot of people, when they hear the term adult stem cells, they're thinking of something that's real old and but it just means that they're not embryonic, right?
Correct. When you're, let's say, the last month of gestation, these stem cells are considered adult stem cells.
Got it. Okay. So they're fully developed, and that's where the term adult. All right. So we have these cells that are basically circulating every day. And explain the physiology a little bit about what triggers them to, be pushed out into the circulation. I thought was interesting that, there's a correlation to circadian rhythm where you'll have more of these in your circulation and later in the afternoon versus maybe before early in the morning. And so talk us through that a little bit, because the physiology here is, I think, very important for folks to understand.
Yeah, because for most people, when we talk stem cells, it's a treatment that you go to a clinic and you get exogenous stem cells. Even if they are your own, we take them out, you put it back in, and once that treatment is done, then you got a stem cell treatment, and that's it. That's the story of your stem cells. And people don't understand that they are in our body since the day we're born, and we release them every day. They have a residence time in the bloodstream of about 1 to 6 hours, average of, let's say, 1-2 hours. So that means that if they're always in your bloodstream, but they reside there for just a few hours, that means you release them all the time. So they're there, and we release them for two purposes. One purpose is repair. So anytime we have an injury, since the day we're born, a broken bone, a cut to the skin, whatever is the injury, or a degenerative process, later in life, anything that is not functioning properly signals. We normally know this signaling with inflammation. Anything that is inflamed is signaling for inflammation, but it's also signaling for repair. These signals, the first of them is going to be released in the bloodstream, will go to the bone marrow, and will trigger the release of stem cells from the bone marrow. It is a response for repair. Now, these stem cells are released after, let's say, a stroke, a heart attack, a bone fracture, a cut to the skin. The increase has been documented to be anywhere between 3 to 10-fold in the number of stem cells in circulation. These stem cells, obviously, when they're released, don't have the knowledge as to where they need to go. What is the tissue calling them? So they circulate everywhere, but the affected tissue will release another round, if you want, of cytokines, of compounds that are going to attract stem cells to that area. So as stem cells go everywhere in the body, when they circulate into the fine capillaries of the affected tissue, locally, they will be able to detect the presence of these compounds, and when they connect with a specific receptor on the membrane of stem cells, it triggers their migration into that tissue, and upon contact with cellular debris of that tissue, they will start to multiply and transform into cells of that tissue. So they are your repair system, and a lot of the studies were about documenting their role in the process of tissue repair. But as these research were done, example, you use a fluorescent stem cell in order to be able to track it in the body, and then you trigger heart attack, and you look at the repair of the heart, and you see the cells of the heart appearing with green fluorescence. So it's telling you that these heart cells were stem cells the day before. But if you let these animals survive, you let these animals live for a while, and let's say 5, 6, 7 months later, you now look at the entire body, and you will find cells that are green everywhere in the body. So basically, out of this emerged the reality that stem cells will go primarily where there's something to repair, but in the background, they go everywhere to simply replace the cells that are being lost. And if I go back to something that you just said before, because I think this part to me is fascinating when you talk about how disease is basically an imbalance between regeneration and degeneration. And that whole concept came from in the very early days when we had the first product, this blue-green algae triggering stem cell release from the bone marrow, the concept that putting more stem cells in your circulation could be meaningful for health. It was just an idea. We had to test it. So I sent the product to a friend of mine in France who had many clients, many patients, and the patients that basically, he was not a doctor, but patients, when their doctor are telling them there's nothing else I can do for you, then they would end up at his doorstep. And so I shipped him product, and I said, give it to all your people and just tell me what you see. And one of the case was a mother who had a 4-year-old daughter with spinal myopathy, genetic disease. She had never walked. She had never stood up. She was crawling some on the floor. The doctor told the mom she would never improve. It's only downhill from now. So she asked me, can I give her Stem Regen, or the product at the time? And I tell her, I don't think it will help because you're going to release stem cells that have the same genetic defect. But if you want to give it to her, man. It's a free country. So
Right.
she gave her the product. 3 weeks later, I get an email. For the first time, she stood up. In
Wow.
another 3 weeks, she started to walk. And then
Hmm.
she was soon at school with the kids. And so the only way that I could explain this case is to think that there is degeneration that is happening, in her case, much faster because there's a genetic defect. But if you can replenish with new cells fast enough to offset the degeneration, then you maintain functionality. So from there came the whole idea. And now there's tons of science behind it. We can spend more time on it, but it's the fact that it's the case for almost the health and the condition of any organs and tissue in the body. Everything is a matter of the normal turnover rate of that organ. It loses cells normally, and these cells need to be replaced with stem cells. But because there's a decline in the number of stem cells in circulation, there's a point where we don't have enough stem cells to offset cellular loss. That is when we start to experience aging in our lives. So stem cells, to go back to your question, do two things. They repair, and they basically replace the cells that are being lost to help you maintain your health as you age.
So give us an idea on how many stem cells, and we're talking about endogenous adult stem cells from the marrow. How many are in a milliliter of fluid or whatever plasma?
In your blood, you have about in the studies that we have done, the average in all the quantification that we have done is about 2.4 stem cells per microliter. So that would be 2,400 stem cells per milliliter.
Okay.
So it's not a lot.
No, but you make a point in the book about the one study where it really only took one stem cell to
Correct.
regenerate the entire hematopoietic system of the mouse, I think it was.
Correct.
Yeah.
Correct. Yeah, because the question was, when you do a cancer treatment, they used to have to find a compatible donor before you go through radiation therapy or chemotherapy. And after the discovery of stem cells in the body, and more specifically, a specific marker on stem cells that allowed to isolate from the bloodstream stem cells, then we were able to collect stem cells from a person, have the person go through their chemotherapy, and then give them back their own stem cells. And so as this protocol becomes more and more utilized, the protocol was to inject 200,000 stem cells per kilogram of body weight. But we need to understand that when they extract these stem cells, it is a very, very, how could I say, a very raw type of extract. Like, you don't know of these 200 stem cells, how many are actually stem cells. I mean, you don't even know if one of them is a stem cell. So the question is that if you give 200,000 per kilogram of body weight, normally it works. So the question that this scientific team had was, okay, well, how many do you really need? Like, if you have only one, but you're sure that it's a stem cells, can it do the job? And they went through a sophisticated protocol that I thought was genius, to be able to isolate one stem cells without touching that stem cells, meaning without marking it with anything to change its nature. And by injecting that one stem cell in an irradiated mouse, they were able in 6 weeks to completely reconstitute the bone marrow, the entire blood system. That mouse was completely normalized with one stem cell. I find it like amazing, the power of the bone marrow.
How did they isolate that one cell? I forgot the way you described it in the book.
I mean, I love when people come up with these like genius protocols. So we need to understand that stem cells, when they are in the bloodstream, if they don't find a place where to go, they will go back to the bone marrow. So the bone marrow is sort of the constant magnet for stem cells. So what they did is that they irradiated female mice, so that killed all the stem cells, and then took stem cells from male mouse, and then injected these stem cells in the female mice. And then 24 hours later, they went in the bone marrow and they did a bone marrow aspirate. Meaning that any cell that you find now in that bone marrow that has been irradiated the day before would be stem cells that came from the male mouse.
Right.
So they took these stem cells and assumed that they were stem cells. So they did this experiment in a number of mice. But the point is that the one that received the one stem cells that was a stem cells, the entire blood system was reconstituted in that mouse. So that's how they were able to have, with a fairly high degree of certainty, the knowledge that that one cell that they isolated was a stem cell.
Yeah, it's very elegant the way they did that.
Yep.
And I think that illustrates how powerful these are, and that, everything's dosage, and, how many stem cells does it really take to affect reparative change, these types of things. And I thought that was meaningful to me, because sometimes we think in these real large numbers, we need millions and millions and millions, and maybe that's not the case.
Well, in this case, we're talking really about the reconstitution of hematopoiesis. Of course, it includes all other stem cells, but when you talk about repair, we're talking about stem cells in the bloodstream, not in the bone marrow. They're released from the bone marrow, so there's sort of two different mechanisms. But yeah, the fact that one stem cell can reconstitute the entire blood system in the bone marrow, to me, it kind of suddenly shows in a powerful way that when you release stem cells from your bone marrow, let's say you have 150 million stem cells in your bone marrow and you release 10 million of them, when you're left with 140 million, you have not put a dent in the bone marrow. Like, it's going to be, it maintains itself at whatever level constantly.
Now, it was originally thought that, I guess they recognized first that these adult stem cells had the ability to regenerate as far as a blood cell lineage, but they didn't really identify some of these pluripotent responses to them. Mm-hmm. Can you break down for the audience a little bit of difference, obviously, between an embryonic cell, which is totipotent, mm-hmm. versus pluripotent, and the difference between, the fact that with an embryonic stem cell, you're likely to get a teratoma, and mm-hmm. not with an. Can you give some background on that for the audience, because it's important?
And let me put that maybe a little bit of context, because stem cells kind of exploded in 2000, and it seems like it started on that day. But there's a little bit of history behind this, with the whole development of the whole concept of embryology, fertility. It's been known for quite some time that life is not like believed, in the 18th century, spontaneous creation. It came because of a sperm, basically coupling with an egg, and then you get the zygote, which developed to a full organism. So we knew that there was such a thing as the first cell that is going to develop into the full organism. And later on in our scientific development in the 60s and the 70s and early 80s, people remember the sheep dolly. People were able to isolate stem cells from the early embryo and really be able to reproduce an entire organism. So we knew the power of these embryonic stem cells to develop into a full organism. So that means they can become everything, and that's why they are called totipotent. Toti means everything, means all.
Right.
So, and really, what was the development of what is modern stem cell research is that until 1998, nobody had been able to grow human embryonic stem cells. They had done chimp, they had done dog, sheep, cat, mice, but nobody had ever been able to grow human embryonic stem cells. So somebody was able to do this in 1998, and it brought back this dream, if you want, to harness this amazing power. If an embryonic stem cells can reconstitute a full organism, that means it can do a heart, it can do a liver, so it could do a new organ for you. That was sort of the dream of what stem cells could do. The problem is that they can do everything. So when you inject stem cells, let's say embryonic stem cells, let's say, I don't know, in the skin, then you're going to get a teratoma, which is some skin, but also some hair, bone, tooth, muscle, a piece of heart beating. So it's a completely disorganized lump of tissue, which basically makes embryonic stem cells completely non-suitable for treatments. So now it's been really used a lot into drug development. We can go into the detail of this if you want, but it's never really be a thing for actual regenerative treatment. The problem is that when the power of these embryonic stem cells was discovered and the way to culture them in humans, there was still this thought or this belief that adult stem cells from the bone marrow can only become blood cells. So they're not good enough. So really we're stuck. We have to find a way to work with these embryonic stem cells. And that's what was so big in the early 2000s, the discovery that adult stem cells are called multipotent, meaning they cannot become everything. But we need to understand the difference is not that great, and the difference means everything in terms of treatment, meaning that an adult stem cells cannot become everything, but it will become the cells of the tissue in which it finds itself. So if an adult stem cells migrates in the liver, it will become liver cells, but only liver cells. And that's what you want. You want them to become the cells of that tissue. So in that way, it suddenly opened the doors to tapping into the power of adult stem cells for various kinds of treatments. It's important to mention also one thing. There's a new type of stem cells that have been discovered. The first discovery was, I think, in 2001, 2003. The first emergence of that type of cells really in the scientific literature, I would say, was probably around 2006, 2007. It has been named different names by different group. The most common name right now is V cells, very small embryonic-like stem cells.
Yes.
And they're calling them embryonic-like because in the human body, they are the cells that are the most totipotent. The most potent, almost totipotent. Like some data suggest that they can even become sperm and egg. They are very small, so when you extract them from the blood, when you do a platelet-rich fraction, then these platelets also contain those V cells. So the healing power of injecting platelet-rich fraction or platelet-rich plasma in a joint, for example, is because of the presence of these V cells to a large extent. So we also have that type of stem cells in the human body that is quite potent.
Yeah, we were one of the first, I believe, in the United States to start using platelet-rich plasma for plantar fasciitis, plantar fasciopathy. And I think we published a small little study in 2001. But it was fascinating to see how you could visualize the tissue changes from, ultrasound exams. Within usually 6 weeks to 8 weeks, we could see this tremendous tissue regeneration. And so I always thought that there was a huge potential for this. Now, one of the problems, the obstacles to that treatment, obviously, was the cost of getting these exogenous stem cells and, whether it was, amnion or however they had processed it. But I thought it was really interesting in your book where you talked about the systemic nature versus the local infiltration, the route of administration, let's call it.
Correct.
And I think you talked about the heart in that as well.
Before we continue with this great discussion, I just want to take a quick break to acknowledge this week's sponsor.
I'm constantly exploring new avenues to enhance well-being and address any health concerns. Today, I'm thrilled to introduce you to an exciting supplement called Stem Regen, which I take daily and genuinely believe could revolutionize your health journey as well. Stem Regen stands out as a game changer in the realm of wellness. Crafted from 5 potent plant extracts, it boasts a remarkable ability to stimulate the release of your body's own stem cells from your red bone marrow. Why is this significant? Well, stem cells are the superheroes of our body's natural repair system. They possess the extraordinary capacity to morph into various cell types, aiding in tissue regeneration and repair. As we age, the quantity of circulating stem cells in our system reduces significantly, impairing our body's ability to heal and rejuvenate itself effectively. Stem Regen steps in to counter this decline by kickstarting the release of stem cells into circulation, thereby bolstering your body's innate repair mechanisms. Once released into your bloodstream, these mobilized stem cells actively seek out areas of damage or injury, initiating the healing process. By incorporating Stem Regen into your daily routine, you're essentially empowering your body to tap into its own regenerative prowess more efficiently. I firmly believe that Stem Regen could be a valuable addition to your wellness regimen, especially if you're seeking to fortify overall health and vitality, accelerate tissue repair, or expedite recovery from injury or illness. This first-of-its-kind stem cell mobilizer is changing the game in regenerative medicine. Don't wait to unleash your body's innate healing capabilities. Visit stemregen.co and use the code SBPOD to get a 15% discount off your first order of Stem Regen.
Yeah, I mean, we are doing a study right now on chronic stable congestive heart failure with a group getting stem regen, a group getting stem cell injection, adipose stem cell injection, and the other one combination of both to have an idea of how they compare. Until that study is completed and published, then we can just suppose on the basis of the data how they compare. But the general comparison to me is when you get an injection, it's a massive amount of stem cells. It can do a lot of good, as you have described. But if you can release them to a smaller extent, but you can do that every day, the infiltration of these stem cells become more physiological, meaning more the way that the body is normally set up physiologically, biologically, to repair. It doesn't repair with a one-time massive influx of stem cells. It repairs normally with small influx of stem cells, which is not to say that it's better or less than an injection. I'm just saying conceptually, that's where I see the difference. But where stem cells have very little access, normally, so where you have very little blood flow, I'm talking here about joints, cartilage, the retina, things like this, then for these conditions, injections oftentimes do much better.
Well, one of the things that I always thought about, I don't know the answer, is, okay, I put these stem cells into their plantar fascia. How long are they really there? I mean, what you see what I'm saying? So after I started looking at your work, I'm thinking, well, if I was going to do one, I would probably want to do both, because I would want the synergy of having the big bolus in the tissue, in addition to continually trying to upregulate their endogenous system.
Correct. No, you're right, because when you inject stem cells in a tissue, I don't think this is very well documented, but a significant number of them simply die. They don't, they get sort of stuck or lodged somewhere in the microvasculature, in the tissue, into interstitial fluid, and they're just there, and they basically die. And as they implode, they release exosomes, which is the communication system of stem cells. And these exosomes are going to really attract stem cells from the circulation to that tissue. And many people believe that the actual repair of tissues come to a large extent from this paracrine effect, the sort of hormonal effect of these stem cells, how they communicate to your stem cells in circulation to migrate in that tissue and coordinate the entire process of tissue repair. We did a study once when we were trying to really better understand the whole process. So we injected fluorescent stem cells, green fluorescent stem cells in mice, and then injected cardiotoxin in the calf muscle, gastrocnemius muscle.
Okay.
And that toxin, cardiotoxin, basically destroyed the muscle, and so that leg was paralyzed. And then we gave them at the time that blue-green algae extract to trigger the release of stem cells from the bone marrow. And what we were looking for is, are we going to see faster repair, better repair compared to a control group? And are we going to see that repair made with muscle cells that are green, meaning that they're derived from stem cells from the bone marrow? And at least in this kind of skeletal muscle, we can say that the entire muscle was green, entirely repaired from cells that were stem cells that basically migrated in that area and turned into muscle cells. The muscle was much bigger, much more developed than in the control group, but it was already there in the control group. So it basically was confirming that stem cells do naturally contribute to tissue repair, but if you can put more in circulation, you enhance the process of tissue repair. So in the case of a muscle, we could say that the stem cells that were released are the cells that contributed to tissue repair. But I know that in the heart, in the fascia, quite possibly, there's the paracrine effect is very significant.
Right. And so when you talked about, I mean, the whole process is very elegant from a physiological standpoint. First, you have these residents in the marrow, and then you have these signaling mechanisms to allow them to get released from the marrow into the circulation. And then you have them migrating to this, let's call it the land of tissue damage. And that you mentioned some of the different signaling mechanisms. So that, and then, there's where VEGF comes in to help get the cell through the capillary wall into the tissue. And then the migration of that cell, once it's made this long journey into, all right, now how does it become a muscle cell, or how does it become a cardiomyocyte? And then you talked about the signaling, the paracrine effect, and microtubules possibly. And so there's different mechanisms that I think you said in your book that still need a lot of exploration to really get a true physiological understanding of what's going on. But the process is really, if you think about it, it's an incredibly complex process that there's a lot of things going on that is going to affect that along the way. And one of the things I wanted to touch on was, all right, as we get older, as with everything, nitric oxide production, testosterone, all of these things, we diminish our capacity to make what we had when we were 20. And the same thing goes with adult stem cells. Our marrow starts going from red to yellow very early, as you point out. And so we're not going to make as much. But if we can mobilize more than are being mobilized, that's a really elegant process in and of itself. And that we'll talk about how your product affects that. But there are certain things still going on in the host that are going to affect the success of this migration. And you mentioned like cigarette smoking. And, so I guess the point is, let's talk about a few of those things, because I think that's going to play a role in how some of our audience is going to want to try to integrate these into their perioperative regimens. But if you have a smoker, that's going to be a detriment. They're not exercising. They're not sleeping well. Stress, you point out. Let's talk about some of those and we're going to talk about how to get these cells mobilized with what you've discovered and brought to the market, which I think is great. But then there's some other things that you're going to want to tell your patient, hey, we need to change these things because they're affecting your stem cell migration, the ability for them to, work with the signaling molecules, all of these things. Can you touch on that a little bit?
Sure.
Particularly the smoking aspect.
Yeah. So first on the conversion of cells into cells of the tissue, very little research the research is there, but nobody has really gone deeply into this whole field. We are still at a phase, it's such new science that we're still at a phase that most of the research that we see is done for treatment of various diseases or by companies or group who gain from the research being developed. Academia has not yet really started to do tons of research with stem cells. So all we can say is that when a cell gets into a tissue, each tissue has very specific molecule specific to that tissue, like nervous system nestin, for example. So when a stem cell gets into contact with protein that are typical to that tissue, I think all it does is that it touches receptors on the surface of the membrane that leads to an intracellular signal to the nucleus that suddenly activates the genes that basically codes for that cell to become a brain cell if it's in the brain. Remember, stem cells have the DNA of the entire body. We all started from one cell. So it's all in the nucleus. So that cell start to now activate its own transformation into cells of that tissue. And but directly to your question about the cigarette smoking, there's a study that we did this was probably 2002, 2003. We had access to patients with Alzheimer's in a hospital. And the hypothesis at the time was, I bet you that we will see fewer stem cells in the patient as people are more and more advanced in Alzheimer's. Now, I must say that reality is true for not almost, all age-related degenerative diseases, except Alzheimer's, where something different has been seen. Some studies have reported that you see the same phenomenon, but others report what we have seen, which is we actually see more stem cells in the blood circulation of many people that were very advanced in Alzheimer's, but the greatest correlation was with systemic inflammation. people had more and more systemic inflammation as they were more and more advanced with Alzheimer's. And what we found is that when we started to look at the phenotype of these stem cells, what kind of receptors and adhesion molecule that they had on their membrane, what we saw is that they had lost the machinery to migrate into tissues, meaning that systemic inflammation had engaged the whole process of cellular migration outside of the fine capillary, because the signaling now has become like it's polluting the entire organism. So the signal to make them migrate is no longer just in the tissue where they need to migrate, but it's anywhere in the body. So so what we found is that, and that's where the idea came, that systemic inflammation is something that can suppress the ability of stem cells to see the tissues that are in need of repair and to migrate into these tissues. It's almost like you don't have enough of a signal to noise ratio. So the stem cells cannot really see where to go and do their work. And as we studied all of this, then I came across other studies talking about other things that can prevent stem cells from migrating and proliferating into tissues. And one of them is cigarette smoking. So they basically would use a pump and actually smoke the cigarette, through a pump, and then take the filter and then dissolve the filter into some water, salted water, PBS, and then would put that in the presence of stem cells. And before and after, you can see that the stem cells move much less in a migration chamber, showing that they have reduced their ability to migrate and also to proliferate. But what was beautiful in that study was that in the full study, quite frankly, I have not seen it published. It was a presentation that was done at one of the meeting for the International Society for Stem Cell Research. And the fellow were reporting that when they filtered the air around this experiment with just the cigarette smoke, and then they took the filter from filtering the air and did the same procedure, it had the same effect on stem cells. So it's not just the smoker, but it's everybody around the smoker.
Mm-hmm. Well, this makes me think back, when you start training and you're working with bone surgery, one of the tenets of that is you never want the patient to be a smoker when you're trying to cut their bone and fuse a joint and get it to heal, because they have such a high incidence of non-union. And
wow, I didn't know that.
I think this is why. I've never heard any reason from a physiological standpoint that says, well, smoking affects the osteocyte, or we just knew that it definitely affected bone healing. But I think this is where that's the tree that we're barking up, and we didn't know. Maybe so I think that's a really interesting thing. It just that the light bulb kind of went off in my head when I was reading your book on that. But I think it's fascinating. So we know that there's power in mobilizing these. I'm going to kind of fast forward a little bit, because everybody that listens to the show knows that, there's the cornerstones of health that you have to have for anything to be optimal, sleep, etc., stress, all of these things. But let's get to how you got to the stem regen product itself, because this is also very interesting.
So when we put on the marketplace the first product, which was just this extract of blue-green algae, it worked very well. We got really, really good stories, good cases. But somewhere along the way, it became clear to me that if we really wanted this concept of endogenous stem cell mobilization, which means the release of your own stem cells, then we needed to have a product that was really, really strong. Really, really strong, so that doctors can use it and really see in their own experience what releasing your own stem cells can do. So this aim of developing a stronger product, coupled with the fact that in my mind there was a question, which is, if we have a plant that triggered the release of stem cells from the bone marrow, and we have evolved in symbiosis with the environment, just like there are many, many antioxidants out there in the plant world, many plants that support the immune system, there has to be many plants that have an effect also on stem cells. Stem cells is new in our knowledge, but when we take a little bit of a step back and we look at it, nothing is new here. We've been healing for thousands of years. tens of thousands of years. So
Millions.
there's nothing new. It's just that now we observe it because we have tools to observe it.
Right.
Before having these tools, the whole phenomenon was invisible. So we have evolved in symbiosis with the environment. There has to be other plants. So how do you find them? So we start to ask the question, what plants have been known historically to be associated with a broad variety of health benefits? For example, like what you have right there behind me, medicinal mushroom. So we studied medicinal mushroom, goji berry for longevity, ginseng, some of these plants, and we found that they all have an effect on stem cells. But we never found like the real like hero plant. Then I went into remote areas of the world, like for example, outside of this global economy. What are they using in Madagascar? Locally, their go-to plant that is good for everything. What are they using in Papua New Guinea, Africa, in South America, Tibetan Plateau? And that is where we found the two most potent plant that we have right now to release stem cells, sea buckthorn berry from the Tibetan Plateau, and one specific species of aloe from Madagascar called Aloe macroclada. So they have 65 species of aloe in Madagascar, but they use only that species, Aloe macroclada, to make a local remedy called the hona that they use for anybody aging, consume that in Madagascar. It's for old age. So we studied these two plants and we found that they were really triggering a significant number of stem cells, release of stem cells, so putting a significant number in the blood circulation. So StemRegen blends the top 5 plants that I have studied over the past 20 years as stem cell mobilizers. So that's really what the product is.
Okay. And from a dosage standpoint, not that we're trying to tell anybody how to prescribe or administer this, but is there a higher dosage recommended for someone who's really depleted and has maybe more chronic disease than somebody who is just wanting to maintain pretty good health from a biohacker standpoint?
Right now, with the science that we have and the numbers that I'm giving you is a really, really conservative number. It's an underestimate just because of the nature of the data that we have. But what we can solidly back is that you take 2 capsules of StemRegen and you will release within 2-3 hours, you will have an additional 10 million stem cells in circulation. So the idea is that if somebody has something that is more significant to repair, then you can take 2 capsules twice or 3 times a day. You could take them all at once. I am sure that it will have a stronger effect than just taking 2. But I think that by experience and also sort of logically speaking, if you take 2 capsules and you will release a wave of stem cells. So you will get better benefits in your body if you release 3 of these waves in a day instead of a larger one once a day. So what we do in our studies, like in the study on congestive heart failure with another study on Parkinson, so we're doing 2 capsules 3 times a day.
Okay. And I would think that if we were wanting to integrate this into a perioperative regimen to optimize that patient for their surgery and for their recovery, we would probably want something more on that type of a route, or excuse me, that type of a administration where it'd be maybe TID or BID versus a single dose in a day.
Correct. Nothing in your body is going to scream for repair more than an open wound. So a surgery is a controlled wound. So it screams for repair. So if you don't have enough stem cells in circulation, the local fibroblasts are going to kick in and you'll start to heal that wound with cells that are going to create a scar, adhesion, the kind of stuff that you would say normally in somebody that does not repair very well. If you put more stem cells in circulation, they will all be attracted naturally to that area, and your stem cells will become muscle cells, tendon, keratinocyte, hair follicle, sebaceous glands, sweat glands. I mean, everything that is normal to the skin and to the local tissue. And we have seen, we're working right now with orthopedic surgeons, for example, and what they see is phenomenal. You just put more stem cells in circulation and you don't have to do this for very long. So you would do exactly what you described. You do this 2 or 3 times a day, you don't have to do this for more than 2 weeks, and you will clearly and very quickly see the enhanced ability of the body to repair when you give the ability of the body to put more stem cells in circulation.
That's beautiful. Yeah, I think there's a lot of application for this, and hopefully there'll be more studies, from an orthopedic aspect as far as bone healing and that. But Well, before we sign off, I want you to tell everyone how they can get in touch with you at stemregen.com, right?
Stemregen.co. So
Okay.
.co, correct. And
Okay.
so the product is there, and also the book is there. The book is really, I know it's a longer read than a small article, but it really gives the whole story in the background, because I think that oftentimes when I talk about stem cells, people still do not fully comprehend the unbelievable power of our own stem cells. I mean, we have studies showing how it can completely regenerate the heart and other tissues. So your stem cells are really powerful, and that's really what I wanted to convey into that book. So both of them are on the website.
Well, I think anybody from a clinical standpoint, anybody who is playing around with peptides or playing around with, amnion, chorion, Wharton's jelly, all of these other things, this book is what I would consider a fundamental text for really understanding, even though you're not necessarily dealing with those other modes of stem cells. But your book, like I told you before, it's really well written, and it's very understandable, whether you're a layman or a physician. And I think it just lays it out, gives you more of a platform to make these recommendations to your patient and optimize their outcomes, which is the goal of every surgeon that I know, is to get the best outcome and get that patient back as quickly as you possibly can.
Correct. And as you said, these other modalities where sometimes we did not really have a full understanding of how they're working, oftentimes they work by leveraging your own stem cells. Mm-hmm. That's really what the mechanism of action is. So by putting more stem cells in circulation, the two of them have a powerful synergy.
Yeah, I think the take-home message here is, well, there's a lot of take-home messages because you've got an incredible body of work, but I think one of them is that don't discount the natural regenerative capabilities of the human themselves, and being able to upregulate that component in combination with all of these other things, I think is a really good place to start. If you want to get this integrated into your patient management, this book is pretty critical because it'll help you understand it. Even if you have a pretty good grasp of this technology, I think it helps you understand it. And if you understand it a little bit better, then you're able to explain it to patients a little bit better. And when patients understand things, then they're more likely to integrate that treatment or that mode into their overall treatment plan. So I think it's really great. And I want to compliment you on that book because you did a great job on it. I could tell you were an educator and worked with people and getting them to learn faster because, I sometimes have a difficulty getting through these books and everything like that. But this was like I couldn't wait to get back to it. It was really great. Christian, anything else that you want to say before we jump off? I really appreciate you taking the time to do this. I'm going to try to really start integrating this into our practice, not only that, but my own self-care. So appreciate it.
Yeah, we covered a lot of material. I would say the only thing to just bring back as a conclusion is to understand it's not just a small thing or an additional thing in the body to know beside other thing. I think that the real discovery here is that it is the fundamental health system of the body. The next book that I'm working on is to talk about the link between the number of stem cells in circulation and health and aging and disease formation. And it's to understand that this notion that we have normally in medicine, that we age because our organs and tissues are aging. So when you have a liver, then you have a liver now that is 40 or 50 or 60 years old. And it's not the case. What stem cell research has shown is that all the tissues are constantly in turnover. You get a new liver every 2-3 years, 4 years, new muscle every 9 years. The time frame is not clear because little research has been done to clearly define this. But the thing that is obvious now is that everything is in turnover. So although we look like we're just moving in life and we're aging and we are not aware that what our stem cells are doing, they are what allows your organs and tissues to keep functioning as you age. They are it's massive what they do in our body. And I think to me, that's the key message. It's not just a little thing. It's like the fundamentals of our health.
Yeah, it's how we continue to exist. It's how we repair. It's how we heal that bone cut or that laceration. Yeah, no, it's great stuff. Thank you so much. I really appreciate it.
I was sharing this message. I was sharing this message recently with a friend, because we don't oftentimes think of it that way, but you probably cannot live without a heartbeat for more than a few minutes. You probably cannot live without oxygen for more than maximum 10 minutes. You cannot live without water for, let's say, more than 2 weeks, food more than maybe 6 weeks. You cannot live without stem cells more than 12 weeks. It is that vital to our survival.
Right. Well, and like you said, since humans started walking the planet, those stem cells have been doing their deal. And
exactly.
They were just, so great. I appreciate it. Really excited to get started personally on your stem regen. And thanks so much for coming on.
My pleasure. Thank you.
Have you been diagnosed with a Morton's neuroma? Are you suffering from persistent pain in the ball of your foot? Does it feel like you're walking on a pebble or a rolled up sock, even when there's nothing in your shoe? You might be experiencing Morton's neuroma, a common but often misdiagnosed condition that can significantly impact your quality of life. But there's hope. The doctors at U.S. Neuropathy Centers, under the direction of Dr. Stephen Barrett, the inventor of this endoscopic technique, offers a revolutionary solution: in-office, minimally invasive endoscopic treatment for Morton's neuroma. For years, patients had limited options for treating Morton's neuroma. Traditional surgery often meant long recovery times and potential complications. But with our advanced endoscopic technique, we're changing the game. Unlike traditional open surgery, U.S. Neuropathy Center's minimally invasive approach means pinhole incisions, less pain, faster recovery, minimal scarring, quicker return to normal activities. Our goal is to get you back to doing what you love, pain-free. With this procedure, most patients experience significant relief and improved mobility. Don't let foot pain hold you back any longer. Experience the difference with U.S. Neuropathy Center's minimally invasive endoscopic treatment for Morton's neuroma. Take the first step towards a pain-free life. Contact us today for a consultation. Call now 404-208-9892 or visit our website. Your feet will thank you.
I hope you all enjoyed today's show and got some truly empowering knowledge out of it. You can always follow up on anything we talked about in the show notes found at our website, podofinquiry.com. If you think you might have tickled just a few neurons inside that cerebral cortex, please spread the word to your friends and colleagues and tell them that they need to get spelunking. Also, if you can take a minute to leave a review on your podcast provider of choice, and especially Apple Podcasts, that would greatly help the show and be so appreciated, provided it's positive. And if not, then just keep your pie hole shut and stay down in that cave. Let's keep spelunking.
This podcast is designed for informational purposes only and does not constitute any medical or surgical consulting advice or imply a development of any physician-patient relationship. The opinions of guests who are featured on the show are not necessarily the opinions of Dr. Barrett or the production team. This podcast is owned solely by Barrett Medical and Surgical Media, LLC. While the show is highly oriented for physicians and healthcare providers, anyone interested in the improvement of human performance and understanding will find this a welcome goblet to sip from, or guzzle. However, no representation or warranties are made in any way whatsoever on this podcast for any products, techniques, or other things discussed. Invited guests are not vetted by the Pod of Inquiry for their qualifications and may have a direct or indirect financial interest in what they present and discuss on the show. The Pod of Inquiry disclaims any responsibility from anything taken from the show and used personally or professionally. It is a responsibility of the listener to perform their own due diligence prior to the implementation of any ideas, products, techniques, or anything talked about on the show.